When geneticists compared DNA samples from modern Australians and Germans, they expected to confirm what anthropology textbooks had taught for decades. What they found instead challenged everything we thought we knew about human migration, genetic diversity, and the very concept of racial categories. The results were so unexpected that 3 different labs had to verify the findings independently. And when the data finally went public, it sparked a scientific controversy that's still reshaping our understanding of human genetics today. This is the story of what happened when hard science collided with centuries of assumptions about race, ancestry, and what it means to be human. If you want to understand where science is taking us next and how genetic research is rewriting human history, hit subscribe. Next week, I'm breaking down the newest findings on Neanderthal DNA in modern populations and why some of us have more of it than others. You won't want to miss that. But first, let's talk about how we got here. For most of the 20th century, scientists categorized human populations into distinct racial groups based on physical appearance. Europeans were one category, Indigenous Australians were another. The assumption was simple. Different continents meant different evolutionary paths, which meant fundamentally different genetics. This wasn't just abstract theory. It shaped everything from immigration policies to medical research. Doctors prescribed medications differently based on race. Governments justified segregation using scientific language about genetic purity. The whole system rested on one foundational belief: that human populations were genetically distinct in meaningful ways. Then DNA sequencing technology arrived. In the 1990s, researchers finally had tools precise enough to compare genetic material across populations. They started with small studies, comparing specific genes between groups. The results were interesting but not revolutionary. Europeans had certain genetic markers, Africans had others, Asians had their own patterns. These early findings seemed to confirm what everyone already believed. Different populations looked different. And now genetics was showing they were different at the molecular level too. Scientific journals published papers about population-specific genetic variants. Medical textbooks added chapters on racial differences in disease susceptibility. But these early studies had a critical limitation. They looked at tiny fragments of DNA, not the whole picture. It's like judging 2 books by reading one paragraph from each. You might notice some differences, but you're missing the full story. The technology of the time could only sequence small sections of DNA at a time. A typical study might examine a few hundred base pairs out of the 3 billion that make up the human genome. That's 0.00001% of the total genetic code. Drawing broad conclusions from such limited data was like trying to understand an entire movie by watching 3 random seconds. Worse, researchers often chose which genes to study based on assumptions about what would show differences. If you're looking for genes related to skin color, you'll find differences in skin color genes. But that doesn't tell you anything about the rest of the genome. Everything changed in 2003 when the Human Genome Project completed its work. For the first time in history, scientists had a complete map of human DNA. Now they could compare entire genomes, not just fragments. The full story was finally readable. That's when researchers decided to do something ambitious. They would collect DNA samples from populations across every continent and compare them comprehensively. No more fragments, no more guesswork, just complete genetic profiles compared side by side. The team assembled was international. Geneticists from Australia, Germany, the United States, and a dozen other countries. They needed samples from indigenous populations who had lived in their regions for thousands of years, plus samples from populations known for extensive migration and mixing. Australia and Germany became focal points for an important reason. Indigenous Australians represented one of the most isolated human populations in history. Their ancestors arrived in Australia somewhere between 50,000 and 65,000 years ago, then remained largely separated from other human groups for tens of thousands of years. If any human population should be genetically distinct, it would be them. Germany, on the other hand, sat at the crossroads of Europe. Centuries of migration, conquest, and trade meant German DNA represented a genetic melting pot. Celts, Romans, Slavs, Vikings, and countless other groups had all left their mark on the German gene pool. The hypothesis was straightforward. Indigenous Australians should show unique genetic signatures, isolated and distinct from everyone else. Germans should show a mixed bag of European genetic markers. The two populations should look completely different at the DNA level. The team collected samples carefully. In Australia, they worked with Aboriginal communities, explaining the research and obtaining informed consent. In Germany, they recruited volunteers from regions across the country, making sure to capture genetic diversity from different areas. Then came the sequencing. Millions of DNA base pairs read, compared, and analyzed. The computers ran for months, processing the massive amounts of data. Researchers checked and rechecked their methods, making sure no contamination had occurred, no errors had crept in. When the first results came back, the lead researcher thought there had been a mistake. The genetic difference between Indigenous Australians and Germans was smaller than the genetic difference between two random Germans. That shouldn't have been possible. These were populations separated by 50,000 years of isolation and an entire planet's worth of distance. The lead researcher, Dr. Sarah Chen from the Australian National University, stared at the numbers for hours. She checked the sample labels. Had someone mixed up the Indigenous Australian samples with European samples? No, the labels were correct. The samples had been handled with extreme care, with multiple verification steps. She ran the analysis again, same result. She brought in her colleague, a statistician who specialized in population genetics. He looked at the data and said it had to be wrong. The probability of this result occurring by chance was astronomical. Something must have contaminated the samples. They brought in a different lab to verify the findings independently. The samples were sent to a facility in Germany that had no connection to the original research. The German lab used different sequencing equipment, different analysis software, different statistical methods. 6 weeks later, the results came back. Same pattern, same shocking similarity. They expanded the sample size, adding more individuals from both populations. They recruited Indigenous Australians from different regions of Australia, thinking maybe the first samples came from a genetically unusual subgroup. They added Germans from Bavaria, from Prussia, from the Rhineland. The pattern held. In fact, it got stronger with more data. Here's what the data showed. If you picked two random people off the street in Berlin, their DNA would differ by a certain amount. Call that amount X. Now, if you compared a random Indigenous Australian to a random German, their DNA would differ by 0.85x. Less difference, not more. The scientific community's first reaction was skepticism. This contradicted everything physical anthropology had established. Indigenous Australians looked dramatically different from Germans. Different skin color, different facial features, different hair texture. How could they be more genetically similar than two Germans were to each other? The answer lay in understanding what DNA actually does. Humans have roughly 20,000 genes. Of those, only a few hundred control the physical features we use to categorize race. Skin color is controlled by about a dozen genes, facial structure by maybe 50, hair texture by another handful. All the rest of our DNA, the vast majority of it, does other things. It builds proteins, regulates cell function, fights disease, processes nutrients. And in those thousands of genes that don't affect appearance, Indigenous Australians and Germans were remarkably similar. But here's where it gets really interesting. Those few hundred genes that do control appearance, they change fast, much faster than the rest of the genome. Evolution is ruthless about optimization. When human ancestors moved into Northern Europe, pale skin meant better vitamin D absorption in low sunlight conditions. Dark skin meant worse absorption, so pale skin genes spread rapidly through European populations. When humans lived in Australia's intense sun, dark skin meant protection from UV radiation. Pale skin meant skin cancer and early death, so dark skin genes dominated. These changes happened in just a few thousand years. Evolutionary time is measured in tens of thousands or hundreds of thousands of years. But these appearance-related genes shifted in the blink of an eye. Meanwhile, the rest of the genome stayed relatively stable. The genes that built hearts and brains and immune systems had already been optimized over millions of years of human evolution. There was no environmental pressure to change them dramatically. The The result is that populations can look completely different while being nearly identical at the genetic level. It's like two cars with different paint jobs, but the same engine, transmission, and chassis. This had massive implications for medicine. For decades, doctors had been prescribing medications differently based on race, assuming genetic differences meant different biological responses to drugs. But if the genetic differences were mostly superficial, that whole approach was flawed. The pharmaceutical industry had built entire research programs around race-based medicine. Clinical trials routinely separated participants by race. Drug labels included different dosing recommendations for different racial groups. The FDA had even approved race-specific medications, like Dill for heart failure in African Americans. The logic seemed sound at the time. If populations had different genetics, they might metabolize drugs differently. Better to be safe and account for these differences than to risk giving someone the wrong dose. But the Australian-German study suggested this entire framework was backwards. Yes, individuals varied in how they processed medications, but those variations didn't track neatly with racial categories. Two people of the same race might respond completely differently to the same drug, while two people of different races might respond identically. The research team decided to test this directly. They looked at genes related to drug metabolism, the ones that determine how your body processes medications. Specifically, they examined the cytochrome P450 family of genes, which handle about 75% of all drug metabolism in the human body. They compared these genes across Indigenous Australian and German populations. They also compared variation within each population. What they found was striking. The differences were minimal between populations. An Indigenous Australian and a German were just as likely to metabolize a given drug, the same way as two Germans would be. In some cases, two Germans showed more variation in their drug metabolism genes than the average difference between the two populations. This meant that decades of race-based medicine had been built on a false premise. Doctors were making treatment decisions based on skin color when they should have been looking at individual genetic profiles. A dark-skinned patient and a pale-skinned patient might need identical medication doses, while two pale-skinned patients might need vastly different doses. The implications went even further into forensic science. Law enforcement agencies had been using DNA to predict race in criminal investigations for years. If investigators found DNA at a crime scene, they would analyze it and announce that the suspect was probably European or African or Asian. Juries heard this testimony and treated it as hard science, as reliable as fingerprints. But the Australian-German study showed how flawed this approach was. Yes, certain genetic markers appeared more frequently in certain populations, but the overlap was so large that you couldn't reliably predict someone's appearance from their DNA alone. The statistical certainty that forensic experts claimed in courtrooms simply didn't exist in the data. A person with mostly European genetic markers might have dark skin. A person with mostly African markers might have pale skin. The correlations existed, but they were weak. Far too weak for the certainty that forensic experts were claiming in courtrooms. Think about it this way. If you found a genetic marker that appeared in 70% of Europeans, but only 30% of Africans, that sounds like a meaningful difference. But it means that 30% of Africans carry the supposedly European marker, and 30% of Europeans don't carry it. If you found this marker at a crime scene, you couldn't say with any confidence what the perpetrator looked like. Multiply this problem across hundreds of genetic markers, and the picture gets even murkier. Some markers might suggest European ancestry, others African, others Asian. What does that tell you about appearance? Very little. Several criminal cases were reopened after this research went public. Defense attorneys argued that DNA-based racial profiling had led investigations down wrong paths, potentially letting real criminals go free while innocent people face suspicion. The scientific community had to reckon with an uncomfortable truth. They had been wrong about human genetic diversity for decades, not slightly wrong, fundamentally wrong. The concept of distinct human races had no biological basis. The genetic differences that did exist were mostly superficial, affecting appearance, but not fundamental biology. and the differences within populations were larger than the differences between populations. Two Germans were more different from each other genetically than the average German was from the average Indigenous Australian. Two Indigenous Australians were more different from each other than the average Indigenous Australian was from the average German. This pattern held across every population comparison the researchers tried. Chinese compared to Nigerians, Brazilians compared to Japanese, Norwegians compared to Indians. Within-group variation always exceeded between-group variation. But if racial categories didn't reflect biological reality, why did they seem so obvious? Why could you look at someone and immediately categorize them? The answer is that humans are pattern recognition machines. We're wired to notice differences and sort things into categories. It helped our ancestors survive. Is that animal a threat or food? Is that plant poisonous or edible? Is that person from my tribe or a stranger? This categorization instinct is so powerful that we apply it even when the categories don't map onto real biological divisions. We see skin color variation and assume it reflects deep genetic differences, when in reality, it's just a few genes responding to sunlight levels. The research team published their findings in 2008. The paper was titled Genetic Structure of Human Populations, and it appeared in the journal Science, one of the most prestigious scientific publications in the world. The reaction was immediate and intense. Some scientists praised the work for finally putting hard numbers to what many had suspected. Others attacked the methodology, claiming the sample sizes were too small or the analysis techniques were flawed. 3 separate labs attempted to replicate the findings. All 3 came back with the same results. The Australian-German genetic similarity was real. Anthropology textbooks began the slow process of revision. Chapters on racial classification were rewritten or removed entirely. Medical schools started teaching that race was a social construct, not a biological category. But outside academia, the message spread slowly. Many people had built their understanding of human differences on the old model. Genetics proved we were all basically the same. That didn't match their lived experience of obvious physical differences between populations. The disconnect between scientific findings and public perception revealed something important about how humans think about race. We don't see genetics. We see skin color, facial features, hair texture. Those visible differences feel more real to us than invisible genetic similarities. This is why the research matters. It forces us to confront the gap between appearance and biology, between social categories and scientific reality. Indigenous Australians and Germans look different. That's undeniable. But looking different doesn't mean being genetically distinct in meaningful ways. Evolution sculpted our appearances to match our environments, but it left our fundamental biology remarkably consistent. The implications for modern society are profound. Immigration debates often invoke genetic or cultural purity. Eugenics movements claim to be preserving superior genetic lineages. Racial supremacists argue that different races have different capabilities. All of these positions rest on the assumption that human populations are genetically distinct in important ways. The Australian-German study, along with dozens of follow-up studies, demolished that assumption. There is no genetic basis for claims of racial superiority. There is no pure genetic lineage to preserve. There are no racial differences in fundamental biological capability. What differences do exist are superficial, affecting appearance, but not ability. And those superficial differences arose recently, in evolutionary terms. Humans spent hundreds of thousands of years evolving as one species. The last 50,000 years of geographic separation weren't enough to create meaningful genetic divergence. This doesn't mean human genetic diversity doesn't exist. It absolutely does. Every person carries unique genetic variations. Some of those variations cluster in populations that have lived together for generations. Scientists can use those clusters to trace ancestry and migration patterns, but the clusters don't align neatly with racial categories. A person might have genetic markers from 4 different continents. Another person might look similar, but have completely different ancestral origins. Appearance and ancestry don't map perfectly onto each other. Modern genetic ancestry services learned this lesson the hard way. Early versions would tell customers, you're 23% German and 18% Nigerian and 12% Chinese. But those numbers were based on comparing customer DNA to reference populations, not on any real biological categories. Later versions got more nuanced, reporting ancestry in terms of regions and migration patterns, rather than racial percentages. The science got better as researchers understood that human genetics doesn't fit into neat boxes. The Australian-German study was part of this broader shift in understanding. It wasn't the only research challenging racial categories, but it was one of the most striking examples. Two populations that looked as different as possible turned out to be nearly identical genetically. This finding has held up over 15 years of additional research. More populations have been studied. Better sequencing technology has been deployed. Larger sample sizes have been analyzed. The conclusion remains the same. Human genetic diversity exists, but it doesn't align with race. The differences between populations are smaller than the differences within populations, and the genes that control appearance are a tiny fraction of the genome, not representative of overall genetic similarity. Where does this leave us today? Medical research has shifted toward personalized genetics rather than race-based categories. Instead of prescribing medication based on whether someone is black or white, doctors increasingly look at specific genetic markers. Anthropology has largely abandoned racial classification, focusing instead on population genetics, migration patterns, and cultural evolution. The field has moved toward understanding human diversity as a continuum rather than discrete categories. But in everyday life, race still functions as a powerful social category. People still experience discrimination based on appearance. Social inequalities still track along racial lines. Legal and political systems still use racial categories. Employment discrimination continues to track along racial lines. Studies show that identical resumes get different response rates depending on whether the name sounds white or black. Housing discrimination persists, with people of color facing higher rejection rates for mortgages and rental applications. Criminal justice outcomes vary dramatically by race, even when controlling for the alleged crime. None of this requires genetic differences to explain it. Social categories can have real effects even when they don't reflect biological reality. Money is a social construct too, but it still determines who eats and who goes hungry. The fact that race lacks biological basis doesn't make racism any less real in its consequences. Education outcomes show the same pattern. Students of color face harsher disciplinary measures for identical behavior. They're tracked into less challenging courses. They receive lower teacher expectations. These disparities persist across income levels, suggesting something beyond economic disadvantage is at work. Health outcomes reveal the impact of social race as well. Black Americans have higher rates of hypertension, diabetes, and infant mortality than white Americans. But these aren't genetic differences. They're the accumulated effects of stress from discrimination, reduced access to quality health care, environmental racism placing toxic facilities in minority neighborhoods, and 100 other social factors. The disconnect between biological reality and social reality creates a strange situation. Science says race isn't real as a biological category, but society treats it as very real, with very real consequences. Some people argue that if race isn't biological, we should stop talking about it entirely. Others argue that we need to talk about it more, precisely because the social construct of race has such powerful effects even though it lacks biological basis. The Australian-German DNA study doesn't resolve this debate. It can't tell us how society should handle race. What it does tell us is what race isn't. It isn't a reflection of deep genetic differences. It isn't a marker of different evolutionary paths. It isn't a biological reality. Race is what humans made it. A categorization system we invented, not one that nature created. And whatever we decide about it moving forward, we can't justify our choices by claiming biological necessity. The genetics tells us that whatever we decide, we can't justify it by claiming biological differences. The science doesn't support that argument. Indigenous Australians and Germans are nearly identical genetically, and so is every other population comparison you could make. We're one species, one gene pool, with minor variations that arose recently and superficially. That's what the DNA says. What we do with that information is up to us.